Yes,
Sure.
forgot.
Yeah, just to type screen.
Yes, it
Um,
started. Nós estamos sendo transmitidos.
but just let me know if my
Fine,
screen
it's fine.
it's fine. It's like a full
Yes.
present. Yeah, okay,
Full
that's
presentation, it's perfect. Yes.
cool. Let's just change
So
for
let's.
laser point.
So, Professor Alexandra Ivo de Medeiros is a very dear friend. She's an associate professor at the Department of Biological Sciences, School of Pharmaceutical Sciences, UNESP, Araraquara. She obtained her PhD in immunology from Faculty of Medicine of Ribeirão Preto, University of São Paulo, in 2002. Her academic journey includes postdoctoral fellowship at the University of Michigan from 2007 to 2009, and a position as a visiting research scholar in the Department of Pathology, Microbiology, and Immunology at Vanderbilt University Medical Center in Nashville from 2019 to 2020. Her research group focuses on the involvement of metabolic pathways in the activation of dendritic cells and CD4 T lymphocytes during the efferocytosis in infection and tumor models, publishing beautiful papers on this subject. Sean and I are the same academic generation, sharing many experiences along our professional journeys. Sean has also participated in several other editions of our course, and we are, again, deeply grateful for her willingness to share her valuable insights with our students. Thanks a lot, Sean, again,
Oh
and the
God,
word is yours.
thank you for your kind introduction. And so it's like such amazing pleasure to be a part of this course. And I totally agree with Dario that you said in reply his email saying, oh, next time maybe you can try to be together.
For
So
sure.
I think, yeah, it's amazing, yeah, because the interaction with the students and also you can do that like in the hybrid way. And so thank you, Ka, Gustavo. It's a great pleasure. Actually, guys, I have learned a lot in the other editions, and what I'm trying to do over here is not just talk about my science, what we are doing in efferocytosis, but also I wanna just share about like all these. Someone is saying something.
No,
Okay.
it's fine.
Yeah, it's just like he tried to give you guys some idea what's going on in all like these different groups that have been working this field that I love, and that's about efferocytosis. And so I want to just start my talk. Uh, oops. So just like for who have never heard about this weird word, efferocytosis, it's kind of like they comes from Latin, like effer means actually take it to the grave with the body. And this is exactly what we're gonna talk today. And so, but what is this grave? This grave in our immunology context is the phagocyte. So this is like just for you guys try to start to understand of what happened actually every single day in your body. So even exactly now in this second, you have like a huge amount of dying cells and in different parts of your body. And so, and this is a homeostatic process, like it happened in every second in your body. And the phagocytes, they are responsible to just clear or just remove these dying cells in your body. And you're going to talk about a little bit more about like how this process works. But in the healthy situation, a healthy body. So you have these cells dying, but you also have in different other scenarios, like not just the cells die every day, but also the other kind of cells. Sometimes I don't know what's working. Yeah. Some other kind of cells in, for example, in some infections. Yeah, you have the recruitment of neutrophils and other inflammatory cells. And these neutrophils, for example, they try to remove and to kill these pathogens as soon as possible. But so these cells now are there and they also need to be removed. So, and this needs to be something like our phagocytes need to remove them as fast as possible, just to avoid some inflammatory or the development some autoimmune disease. So let's just like explain how it's going. I'm planning to explain to you guys how all these scenarios can happen in your body. So you're going to start to talk about the efferocytosis and tissue homeostasis, and then I want to talk a little bit like this is more related that you have done in the lesson decade in our lab. It's about how efferocytosis can help in host defense in different like infections. And in the second part of my talk, and I'm trying to bring to you guys a new, very interesting area that is related to metabolic activation and efferocytosis in different scenarios in cancer as well as in some different infections. So, Ka, I don't know exactly how many. So, it's one like 50 minutes because I saw 2 hours and
1.5 hour and plus 30 minutes to discussion and answer the questions.
okay, that's good.
So
So
feel free.
I just planned like it to like it final like just okay, let's just see how it goes. And I was planning to talk a little bit more. It's even better now. So, guys,
No problem, sure. We can discuss more
okay,
along the
then 1 hour.
presentation.
Yeah, okay. So I don't know, like usually it's better. So you prefer like just keep going talking instead of some students like just stop it and ask some questions, Ka? I want to just make you
So
guys free to, if you have some questions, because some, yeah.
they can raise the hands and do their questions during the talk, but we normally
Okay,
go this
great.
after the talk, okay?
Okay, up to you guys. Okay, so in your body, as I read and start to talk about that in the beginning of my presentation. So you have like in the brain, lung, liver, kidney, and intestine, like a different turnover of these cells. And some of them, for example, gut, you have a huge turnover. It's like much faster, and there's a huge accumulation of these apoptotic cells, and your phagocytes need to work much like faster and if in efficient way. And so just to give you idea, you have over than 200, 300 billion cells dying in your body every single day. This is, they say it's around 0.5 kilogram. And so it's a huge amount of cells dying in your body. And so, and to do this job, you have the professional phagocytes. For example, macrophages and dendritic cells is the major important professional phagocytes in your body, in different parts of your body. But also now we know the non-professional phagocytes, for example, epithelial cells, they also play an important role, and they're not able to phagocyte as much as these macrophages and dendritic cells, but they also really important in this process to keep your body healthy. And so it's the way your phagocytes try to clean or to remove these apoptotic cells every day in your body. It's a very well known nowadays. It's a multi-process process, multi-step processing that in summary, like we summarize in the finding signals. and bind me, eat me, and digest me signals. And so these find me signals, is the time your cells start to release in compounds or mediators that's going to allow your phagocytes to recognize, to be attracted actually in the first step. And so some of them, they are just like chemokines, as you guys already probably know how important it is to promote the recruitment of cells. And nucleotides, ATP, UTP, as well, the ATP degradation products, ADP and MEP, and some lipids. And so I brought like just this amazing paper from Cody from the Rev Labs. Is this paper study have been published almost like 14 years ago. During the time, we don't really know how important it was, this find me process or mediators. But now we know this, when the cells start to die and activate these caspase 3 and 7, the most important in this case, they allow the pannexin channel be activating. And once these channels are activated, they start to release this ATP and also UTP. And so as you can see over here, and when the cells die by apoptosis using the antibody that activating fast molecules, these cells, these apoptotic cells start to release ATP, UTP, but how these nucleotides can be important to promote the recruitment. What they show is if you inhibit using small interference RNA, this channel, this pannexin, you can see a decrease of the release of this ATP as well UTP. But the most important that we conclude like how important they are these nucleotides to promote, to help during the recruitment of these phagocytes is the macrophages, these cell line macrophages, they are not able to be recruited as they are when you do having these ATP and UTP. But even though you still have like some recruitment, that this allows that is not this ATP and UTP play a very important role during this recruitment. We have also all the mediators that are involved in this recruitment. So this for who wants to in the future working with this efferocytosis, I really recommend like all papers from Ravi Labby, and they are the pope of efferocytosis as well, Ida, the other group that's they bring like so many knowledge about like efferocytosis in general. So, okay, let's go for the next process. The next step is actually the recruitment and the binding or the recognition of these apoptotic cells. And so, of course, and to allow these macrophages and dendritic cells be recruited, they have all the receptors can recognize in these mediators, yeah, and this is exactly what we are showing over here. But after being recruited, we needed to differentiate like two different kinds of cells, yeah, in this context of efferocytosis. So your phagocytes need to recognize in which cells they're going to phagocytes. And as you can see here, your healthy cells, you have the expression of these molecules that we call don't eat me signals. And this is really important, you know, like, because when you have like this really high turnover, I'm still talking just about during the homeostasis, yeah, in a healthy situation. So your health cells need to make sure they are not going to be recognized. And this is like the way your cells do. They express these don't eat me things just to say your phagocytes, not the time to be removed. However, when your cells start to die, the first thing that is well known is at least is the molecules is well known study nowadays is the exposing the outer plasma membrane, the molecules that we call phosphatidylserine. And phosphatidylserine is like the molecules that can be exposing in your apoptotic cells, and that allows your macrophages or dendritic cells as well, epithelial cells being recognized. And as you can see here, if I give this talk like 25 years ago, in that time, we believed there was just like a phosphatidylserine receptor. We didn't know we have like this huge amount of options to recognize the phosphatidylserine in the surface of your dying cells. And as you can see here, you have some receptors that can directly recognize this phosphatidylserine. However, you also can what can happen is some bridge molecules can also help to make this process even easier. Like we call like a kind of opsonization of these apoptotic cells. And these bridge molecules can be released from your phagocytes and also some from apoptotic cells. And so these different receptors, they have this really important job, and is like just to make sure your tissue is going to be safe. And so what I want to mean about exactly about that. So sometimes they don't. Okay. So what does it exactly mean in terms like of this science in the tissue that is you have like all these apoptotic cells in your body. So we have this just the touch, between the phosphatidylserine and these efferocytic receptors. They allow to prevent, for example, the expression of TNF, and some of these receptors is MERTK that we call in general like TAN receptors, and they not just like impair the expression of TNF, but also the touch of these receptors between in phosphatidylserine allows to increase the SOX that impair the STAT, like basically to be activating. Moreover, we have other TIM receptor and stabilin, for example, they inhibit the activation of NF-kappa B that impair the TNF, IL-6, and CCL5 being secreted for phagocytes as well. The same, and stabilin too, they have like this capacity to induce the production of some anti-inflammatory mediators such as TGF-beta. So basically what I'm talking about, it's a very anti-inflammatory environment. And so this is the reason, even though you have like this huge amount of cells dying in your body, you don't have you cannot even notice it. So if you do like hematoxylin and try to identify that, so the accumulation of these cells, this process is so efficient that you cannot even identify these cells dying in your tissue. So the conclusion is like if you don't have a very efficient process of these cleansing of these apoptotic cells, you can trigger what we know much better now, some autoimmune disease or inflammatory diseases that is basically associated of some defect of the cleansing of these dying cells, and this can promote the tissue damage. So some of these autoimmune diseases related to neurological disorders, for example, Guillain-Barré syndrome, or lupus, that is very well known now. The defect it's a very compromised defect of these dying cells, and arthritis and Crohn's diseases. And I just brought a study that it's associated to arteriosclerosis diseases. And this is because like these two studies, they show these very elegant results. What they observed is in the arteriosclerosis that can make the individual to have a heart attack or a stroke. They have like these two studies, they come like one after other in 2016 and the other one in 2017. And one of them, they show in these patients that have this arteriosclerosis, actually the cells, they express this don't eat me signals that I just told you guys. and they have much more CD47 expressed in these cells. And as you can see here, they identify in patients that they have this sclerosis, this arteriosclerotic plating. And you can, as you can see here in red, different than patients like healthy individuals, they don't have the expression of CD47. But as you can see over here, there's a huge accumulation of these don't eat me signals. And this causes like a very accumulation of necrotic cells. I didn't bring these results, but the other association they have done in this arteriosclerosis disease is the TNF is the most important mediators that it's activating these apoptotic cells. Actually, the cells, they don't become apoptotic. And this allows the cells keeping expressing as like mimicking a healthy cells. And the problem is if once these cells express these don't eat me signals, they are not removed, and this causes these necrosis. And when you have necrosis, you have inflammation, and this can be the reason in these patients like to promote this heart attack and strokes, just because you have, instead to remove these apoptotic cells in this in this plate, and they keep just living there forever, and this promoting the accumulation of these necrotic cells. But this is like a paper came 2016. After 6 months, the ARA Tabas groups, they published this amazing work in showing, first of all, there was this correlation of the accumulation of necrotic cells and also soluble MERTK receptors. Just remind what I told in the beginning of my presentation. What they observed is in these patients that they are more susceptible to arteriosclerosis, they have the high expression of these metalloproteinase that we call ADAM17. And what this metalloproteinase does in the cells, they basically cleavate these receptors that is really important to remove these, recognize these apoptotic cells. And for this reason, we have this accumulation of these soluble MERTK receptors. Instead to be in the membrane to remove these apoptotic cells, they are released. And this is like in the symptomatic patients, we have this accumulation of the soluble MERTK, and also you different than asymptomatic individuals that you see the MERTK expressed in the membrane of the cells, as you can see over here in red. In patients that they have more susceptibility or they are developing these arteriosclerosis, you don't see as much as in these asymptomatic individuals. And what they try to prove how important is the efferocytosis in this process. They use this model for arteriosclerosis using this mice that they basically they don't express this receptor are important to remove the LDA in lipoprotein, that is the bad one, in your body. And so what like they basically did is they transfer the bone marrow from mice. They are resistant to this metalloproteinase. So this means that this mice that is already going to develop the arteriosclerosis, now when they have this new bone marrow, the cells, the phagocytes are going to express again the MERTK. And what you can see when they do that, we don't have anymore the soluble MERTK receptors, and you see a very pronounced decrease of the accumulation of these necrotic cells. So this is one example how efferocytosis can help you to avoid, for example, to die by heart attack and stroke, and if you have this defect to remove your apoptotic cells. So in summary, if you have some defect of some receptors, or if your cells, apoptotic cells, your cells keeping expressing some don't eat me signals, you probably going to have some problems because you have a high chance to develop some autoimmune disease, as I just showed you guys. So the second part is not the second part actually. The next scenario I want to introduce to you guys, it's about what I have done actually in our group. So as I just showed you guys, like most of the studies related to efferocytosis is more related in this scenario that we don't have in the infection. And when I started like in my group in Brazil after my postdoc in Michigan, in Mark Peters' Golden Lab, who's a professor that has been working with EPD too, but he never have worked before with efferocytosis. But so we have a question that time that in patients that develop COPD, they have like the accumulation of these apoptotic cells. And my job over there is try to understand how efferocytosis and or the mediators produced in this situation could help in some way to avoid or to promote these anti-inflammatory mediators. But when I came to Brazil, I want to just do something different. And I decided to start my lab asking different questions, not like in a just inflammatory disease. I want to understand what happened when the pathogen are inside of these apoptotic cells. So as you know, during the infection, as I told in the beginning of my talk, there is a huge recruitment of neutrophils, for example, to try to remove the pathogens in the tissue. And the dendritic cells are responsible to degrade these apoptotic cells that they can have or not, like some pathogens. And these dendritic cells, when they phagocytes, in this case, apoptotic cells contain the pathogens, they start to release a different kind of mediators. And as you know, this microenvironment is very important to promote, to drive the T cells differentiation. So our question is, once you have like apoptotic cells that I just told you guys, they basically release anti-inflammatory mediators to keep your tissues healthy. But when you do have the pathogens inside of these apoptotic cells, the question was, how these dendritic cells handle these pathogens and promote the differentiation of T cells? So the efferocytosis like really can help the clearance and can help it to promote, to drive the response in during the situations. And so this is basically what we have done in our last decade. We try to understand in different infections, for example, Staphylococcus, Streptococcus, and recently in Zika virus, if those pathogens inside of these apoptotic cells can drive a different like T cell differentiation, or really can help into clearance, to help in doing the clearance of this infection. So what you have done, and I just try to summarize in our results in this cartoon, is when dendritic cells, phagocytes, no infect, or basically it's exactly what I told in the beginning of my talk. When you don't have the pathogen inside of your apoptotic cells, your dendritic cells just drive a very anti-inflammatory microenvironments. And what we have done, we have observed, is this microenvironment produced by dendritic cells, they basically drive it to Treg cells. That is very fair, yeah, because we don't have any kind of other compounds inside of these apoptotic cells that can drive anti-inflammatory. There is no reason to produce these anti-inflammatory mediators. And the reason is pretty much what I told you guys. They recognize phosphatidylserine, and when these corpuscles are degrading this phygolysosome, there is no pathogen to bind in any toll-like receptor, for example. So this drive a very anti-inflammatory microenvironment. However, when you have infection, for example, by E. coli, it's a gram-negative bacteria. What we observed is when the dendritic cells recognized by phosphatidylserine, even though they still recognize by phosphatidylserine these apoptotic cells, when those cells are degrading this phygolysosome, they can promote the activation of some toll-like receptor. In this case, was toll-like receptor 4 that disallow the activation of NF-kappa B. And different than observed in this non-infected context, we observed the dendritic cells producing these very inflammatory microenvironment that drives Th17 differentiation. And so the other very important compound that we already know that play a very important role during the differentiation, it's a lipid mediator that called PGE2. And so PGE2, it's well-known lipid mediator that can help in T-cell differentiation in the general, but they never really, there are no studies showing that the PGE2 can help during efferocytosis to drive this T-cell differentiation. And as you can see over here, the PGE2 also can help to drive these Treg cells. And as well, the PGE2 can also help in Th17 differentiation. But so in this situation, even though you have PGE2 in these two scenarios, you also have the cells also produce all these inflammatory cytokines. So PGE2 in these two different contexts can drive different like inflammatory T cells and or suppressor T cells. But so always when we are in present our data in a meeting, so the next question was always, what about if you change the pathogen? Do you think like you're gonna have exactly the same behavior during the efferocytosis of these infected apoptotic cells? And the answer is no. Even though you have a PGE2 in apoptotic cells infected by, for example, Streptococcus pneumonia, as you can see over here, the pattern or the T cells are driving in this scenario, it's totally different than E. coli infection. You have like most T helper 1 being differentiation in the presence of the products like it released from dendritic cells, in this case IL-12, TNF, and TGF-beta. And the amount of PGE2, it's like not even close the amount of this prostanoid in the context of E. coli infection. So in the PGE2 in this scenario, when you have a Streptococcus pneumonia infection, has played no roles during the Th17 differentiation. So this means that PGE2, even when they are there, they not necessarily are important to promote the differentiation of these inflammatory T cells. And the lesson, but not least, infection that we are working on in the last 2 years, it's in Zika virus infection. So I don't know if you guys know, during the infection by Zika virus, you have a huge accumulation of apoptotic cells. And our question actually was not related to microcephaly that so everyone probably have heard about during the Zika infection virus. But what there is a very strong correlation associated to when you are infected for Zika virus, you have a chance to develop some autoimmune disease, for example, Guillain-Barré syndrome. And so our question in this project is try to understand if during the efferocytosis of these apoptotic cells infected with the Zika virus, they can promote the activation of these autoreactive clones. And the idea was some studies from Blender's groups, Julie Blender, they already show some gut infection. They can promote the activation of Th17 cells that they recognize the self-antigens. And for the reason for that, if you think about doing the degradation of these apoptotic cells, they have in this case in the Zika virus, when your cell is going to present these products from the degradation of this body, you present like a self-antigen as well the compounds from the virus. And the idea is this can you increase the chance to promote the activation of these autoreactive clones. You are still working this project. This is the reason I didn't bring like the full results. And you have a strong evidence that when you have a high rate of Zika virus infection, you increase the chance to promote the activation of these, as we know so far, Th17 cells that can recognize some self-antigens. So the other part of this talk, it's tell a little bit about the metabolic changes that occurs during the efferocytosis. So this area, it's pretty much new, and I would say in the last 7 years, no more than that, I think. It's the major studies that have just bring like all these amazing results that showing that efferocytosis can also change the metabolism of the cells. So just a brief introduction. So the metabolism so we think like, let's say 15 years ago, or maybe a little bit more, was just responsible to producing energy. But in the last decade, we have understand that the metabolism can also play a very important role to promote the activation of different cells, not just T cells, as well macrophages and dendritic cells. And so just to summarize in a very brief way, how this metabolic pathway can sustain the cellular activation. So what we know, and this is not like it's not even fair to summarize in this way, but just to try, because new studies change a little bit this information, but in general, let's say in this way, when you have anti-inflammatory macrophages as well tolerogenic dendritic cells, beta oxidation and also OXPHOS, they are the one play an important role to maintain these cells quietened. And in T cells, for example, Treg cells, also beta oxidation play the most important role to keep to maintain the phenotype of these Treg cells. And so, however, when you have the activation of those cells, the pro-inflammatory macrophage and the immunogenic dendritic cells, the glycolysis and fat acid synthase, as well pentose phosphate, but glycolysis and fat acid synthase, they are the one play an important role to promote the activation, to help the activation of these myeloid cells. And as well, for T cells, Th17 and T helper 1 cells, glycolysis and as well glutaminolysis are the most important metabolic pathway that sustain or to activate and sustain these phenotypes. And so what the way we try to study, understand like how these different metabolic pathway can really help and that during the activation of these cells, it's basically to using, of course, it's not just pharmacological approaches, but it's a good start to understand how these different pathway can really be involved in the activation of these, in this case, in our case, we are interested in dendritic cells. And so I'm just showing guys, because we are going to talk about like this these drugs or this pharmacological approach that you can use in your study, try to identify how this metabolic pathway can really play some important role in doing this whatever cells you are interested to look in your model. So, but in our case, what we are really interested to understand is how during the efferocytosis, this metabolic pathway could play important role during not just during the homeostasis, but also during in our interest in, for example, infection as well as in a cancer, like in the tumor microenvironment. And so everything I'm gonna tell you guys in the next few slides, it's about macrophages, and macrophages is the most phagocyte has been studied in efferocytosis in general, and we are interested in dendritic cells for different reasons that I'm gonna tell you guys. But so macrophages are very inspiration for our group, just to follow what the study in efferocytosis can help us to understand more in dendritic cells. So I just brought, if you want to know a little bit more about like metabolism during the efferocytosis, I really recommend this review in Cell Metabolism that has been published in 2021. What they know is, remember, like when I told you guys during the efferocytosis, we have this multi-step process that we call the find me signals, the binding, and as well the internalization, the phagocytosis, and also the digestion. Now we know in all different steps of this processing during the efferocytosis, they already can start to reprogram these phagocytes to change the metabolism of these cells and help these cells to prepare themselves to degrade this apoptotic corpus. And so, as you can see over here, some of these information I already have done in the beginning of my talking, but I want to just give more information about that. So remember the find me signals that so your apoptotic cells start to release as soon as these pannexin channel are activated by caspases 3 and 7. These metabolites, they can not just start to attract the phagocytes, but as you can see over here, they allow through the receptors, yeah, they bind in these phagocytes. They allow some change in terms of the transporters. That's going to be very important to prepare your phagocyte for the next step. For example, some of these transports is GLUT1. You know, when these phagocytes start to interact with these metabolites that your apoptotic cells are releasing, these cells, these phagocytes, in this case, macrophages, that it's the most important over here, start to express some GLUT1, for example. GLUT1, it's a very important transporter to captain, like just captain this glucose from outside of your cells to inside of this phagocyte. And this is the first step to start the glycolysis activation of this phagocyte. In the second step, when your phagocytes start to touch into phosphatidylserine or to recognize this phosphatidylserine in the surface of the apoptotic cells, as you can see over here, it occurs like a very important processing. They started to increase the transporters of some cholesterol. But what the reason for that is, what they say is, they are preparing your phagocyte for what's coming. Just remember, your apoptotic cells, they have a lot of lipids. And these macrophages, it's so necessary, they need to manage and to remove this huge amount of lipids that they are going to keep inside of these phagocytes in few minutes, in few hours. And so they increase the transport of these cholesterol transporters, and also start to increase the glutathione to make sure the glucose is going to be able to get inside of the cells. And the next step is the internalization and degradation. And I'm going to show you guys in a few seconds. And in addition to increase the transporters of cholesterol, they also increase the transport of the esterols. That is also the compound is going to be increasing inside of this phagocyte, during the degradation. And so they to avoid these cells to be had a huge amount of the cholesterol inside, they started just to remove it or just to take it off all these huge amount of the lipids inside of the cells. And the other transporters that it's really important is the amino acid exporters. And you're gonna see that inside of this phagolysosome, they transport this amino acid that is going to be very important to keep these phagocytes just keeping doing several rounds of the efferocytosis. So this is just to summarize that in all steps of the efferocytosis process, the metabolism can also play a very important role, basically to prepare those phagocytes to receive and to degrade these apoptotic cells. And so this is a few studies so I want to show you guys in related to metabolism and efferocytosis, and what and just to tell you guys how everything in terms of efferocytosis and metabolism is pretty new. This is one I'd say the most important papers that came out in 2019, and in the last, as I told you, the last 6 years. And what they observed is, so we already know the IL-10 and TGF-beta is the most important anti-inflammatory mediators that are produced during the efferocytosis. But what we never really think about is the metabolic pathways could play an important role during this process. And this is what the metabolism field just brought to us in our studies. And what they observed is the efferocytosis induce the production of IL-10, but what we never think about is the beta oxidation was involved in this process. So during the degradation of these dying cells, remember, as I told you guys, there is these transporters of these lipids in the cells during the efferocytosis. And so these lipids, they can be they are being released from these phagolysosomes, and you have some transports that can just allow these lipids comes inside of the mitochondria. And what this paper show is this beta oxidation, they play, just to make a long story short, and they are responsible actually the transcription of this IL-10 and promote all these anti-inflammatory mediators. And this paper is related to cardiac attack. And so the macrophages in your heart, and they remove all these cells when you have a heart attack that die in your heart, and your macrophages started to produce all these IL-10 to promote the resolution of the inflammation after the heart attack. And so the other paper that just brought us a very important acknowledgement is during the efferocytosis, the other compounds are produced and play an important role is the lactate. So during the efferocytosis, we already know there is a increase of the glycolysis, and the glycolysis, they promote the production of a huge amount of lactate that can be transported, can be staying inside of the cells, and the accumulation allows to transport this lactate outside of the cells. And now we know the production, this lactate being released in the microenvironment, in this microenvironment, promote the M2 phenotype or anti-inflammatory phenotype of those macrophages. And we already know when you have M2 or when you have like these anti-inflammatory phenotypes, those cells are much more capable to remove other apoptotic cells. So this means that the efferocytosis not just increase the glycolysis, but the production of lactate just make the other macrophage is more able to remove the apoptotic cells. And the other very interesting paper related to efferocytosis and metabolism, from Ira Tabas, what they observed is during the efferocytosis, you have increase of these transporters that allow the amino acids such as arginine being just released in the cytosol. And the arginine and also ornithine, we know like they promote the activation of RAC. And this RAC, it's very important molecules involved in the phagocyte of these apoptotic cells. So the idea is in the first round of efferocytosis from your macrophages, when the apoptotic cells are degraded inside of the phagolysosome, the arginine that are basically transported to the cytosol, keeping your macrophages phagocytes like more and more and more apoptotic cells. So this is the round, is the capacity of the different round of efferocytosis is related to this metabolism, amino acid metabolism, so it's play an important role to maintain your body healthy. And the last two studies that I want to show you guys, it's about like the PPAR gamma, that so we know during the efferocytosis, the expression of PPAR gamma, it's increased a lot, but this we already know. What we didn't know is the lipids were able to bind in this PPAR gamma and induce not just the IL-10 and TGF-beta production, but also they increase all these efferocytosis receptors, efferocytic receptors that keep your cells just like doing like different rounds of these efferocytosis. So this is like to summarize like what we know nowadays about the metabolism being playing an important role during the efferocytosis. And so what I want to show you guys what we have done in our lab in terms of metabolism and efferocytosis. And the focus of this part of our study is basically in tumor cells. So everything in our lab starting when we started to be interested in cancer when I went to Jeff's lab in Vanderbilt University, as Cass said in the beginning of my talk. So I was looking actually for another model. I was kind of like boring to work in just in infection, and I wanna like just to have like a different like interest in my lab that could like compare all these different scenarios and to understand how the metabolism could play different roles in other scenario than infection model. And Jeff just proposing my first lab meeting in his lab, why you don't work with the cancer? And so I never really was focused in cancer because I think it's a very complicated model to work in. But so, when I just searched for so if there was some studies have been working in phagocytosis in cancer, surprisingly, not like totally surprisingly, because when you have a good idea, always someone already have this idea and is exactly what happened. So this is like a paper and that has been published in actually a decade ago. What they observed is during the efferocytosis, as I told you guys, you have basically a tolerogenic microenvironment, but in terms of dendritic cells, the efferocytosis promote like a very suppressing effect in those cells and macrophages as well. And what they observed if you block in the efferocytosis using an antibody that bind in phosphatidylserine, you impair these macrophages to remove these apoptotic cells. And the idea was basically is in the tumor microenvironment, when you have when we start to treat these tumor cells, there is a huge accumulation of dead cells. These dead cells can be immunogenic cells. We're going to talk about this in a few minutes, but also they can be non-immunogenic dendritic cells. In this case, is exactly when the cells die by apoptosis. And what they observed is this is like a prostate cancer model, experimental model. And as you can see over here, these mice, they receive this antibody that basically blocked the efferocytosis of cancer cells. And the idea is, okay, like if I impair the efferocytosis in this tumor environment, microenvironment, could like I could change like this treatment could change the behavior of these dendritic cells. And what they observed is this is like the group that they just received the tumor. As you can see, it's growing very well. And this is like the group they receive this antibody that are blocking the efferocytosis. And you see like some decrease of the size of the tumor. And this is like the doxytic cell, doxytic cell, I don't know how to say that. And basically is the treatment that it has been used to treat in prostate cancer and working like pretty well. But the animals receive the combination of the antibody as well, the chemotherapy. As you can see, there is a decrease of the size of this tumor. And so this is like the graphic, as you can see very clear. The combination of the antibody that impair the efferocytosis plus the drug has been used in prostate cancer, you have a really good results in terms of decrease of these tumor cells. And so in addition to that, what they observed is that the dendritic cells in this tumor microenvironment, they became like much like immunogenic cells. As you can see over here, dendritic cells can increase the IL-12, TNF production, and decrease of these anti-inflammatory mediators that is very well known that are being produced in the tumor microenvironment. So this can be a good option, like especially for cancers, you don't have a very potential effect, very potent effect to just kill the tumor cells. And surprisingly, this treatment has been actually in some clinical phase, phase 2, as you can see over here, for different kind of cancers, lung cancer, breast cancer, hepatocellular carcinoma, and some of them, they are well tolerated, some of them not that good as for other treatments. And so the idea is pretty much good, like when you think about it, I don't know if this year Lorenzo Galuzzi was a part of this course, but so he works in this amazing field that just showing how immunogenic cell death that release all these damps can be like a very important way to improve the treatment of different cancers, and in using different like chemotherapy that can improve, or make this tumor hot. But so this is like a perfect scenario that in cancer, so if you have like this, if the treatment can induce immunogenic cell death, you are going to have this amazing scenario. So your dendritic cells is going to be recruiting, is going to be activated, is going to uptake this antigen from tumor cells, and they are going to the lymph node to promote the activation of the T cells. These T cells are going to be able to be recruited to this tumor, and everything is going to be okay. So in addition, the drug that can kill directly the cells, or they promote this immunogenic cell death, that also helps to induce to help to to kill these tumor cells. However, you have some treatment that does not work in this amazing way. This cell death can be a non-immunogenic cell death, and you're gonna have an accumulation of these apoptotic cells. And as I told you guys in the last few slides, when your macrophages or dendritic cells phagocyte these apoptotic cells, of course, it's really good during the homeostasis, as I told you, because this can impair to drive in some inflammatory or autoimmune inflammatory disease or autoimmune disease. During the infection, helps a lot when we have the pathogen, because this dendritic cells are going to be activating and help to drive different kind of T helper cells. But in the tumor microenvironment, just think about that. So if you have the accumulation of these apoptotic cells that we already know, it's apoptotic cells that are not allowed because they are not releasing damps as in the immunogenic situation, immunogenic cell death. So this is going to be a really big trouble, in the tumor microenvironment. So what, like, as I just remind you guys, when the tumor cells expose this phosphatidylserine, it's going to happen exactly in the same way that I told you guys during the recognition of these ferrocytic receptors like TIN, TAN, they all of them, this mediator receptor is going to bind, they drive us to this immunosuppression environment. And just think about that happening in your tumor, So, but the concern about like using this antibody that block the efferocytosis, that is a really good idea for sure. This is the reason the studies can now driving for clinical studies. But so just think about that, just following my talk, So when you have like this accumulation of the apoptotic cells in the tumor microenvironment, the idea of this treatment using baviturimax is to impair the efferocytosis, and then it can be a good choice because you're gonna have the immunogenic cell death, And the other option is, once like you have the antibody that can bind in and is going to bind in this phosphatidylserine, now the dendritic cells as well, macrophages, they are going to recognize by EPC receptor, and it's also good. But if you think just in terms of like the tumor microenvironment, so because when your apoptotic cells are removed by FC receptors, different than when they bind directly in the phosphatidylserine, you are going for sure to have a very inflammatory condition, or environment. So, but the problem is this antibody does not like reach or does not are going to be present just in the tumor microenvironment. every single cell in your body, is gonna be impaired to be removed from your phagocytes and all these professional or non-professional phagocytes. And the risk you are going to take is you can develop some autoimmune disease, as I told you in the beginning. So it's going to be, for sure, according to the results I just show you guys, maybe it's going to be very helpful to help you to kill the tumor cells, but otherwise, you have the risk to develop during your life some autoimmune disease, because you are not just impair the efferocytosis of cancer cells. This antibody can also impair the efferocytosis of different cells in different parts of your body. So, and when we just started to think in other approach that could help in the tumor microenvironment to remove these apoptotic cells, because it's going to happen. There is no option, because during the treatment, like, you are going to trigger some of death cells. And so over here, the idea is like, why you cannot like try to manipulate the metabolic pathway of these cells, those cells, when they are removing these cancer cells. So the idea basically is to reprogram these dendritic cells during the efferocytosis of these cancer cells and try to make these cells more immunogenic cells. So you can ask me, how crazy is that? Is that possible? You can really change the metabolism of your immune cells inside of the tumor cells. So this is a very amazing review publishing from the Percy and Edward Percy and Erica Percy couple that works in this field, in metabolism field. And so what they show, we already know that, yeah. So these dendritic cells, they are tolerogenic, your macrophage does not work well, and the T cells, most of them, they are Treg, your CD8 and CD4 T cells, they are exhausted and they are not work at all in the tumor environment. And what they show, what we know, yeah, nowadays, it's all relating of the metabolism of these immune cells. And so in this paper, they just brought the new approach that you can use to try to manipulate the metabolism of these immune cells and try to make these cells more capable to be activated. And the idea is, I just try to summarize and to make you guys understand. For example, the vaccination that we call like the vaccine against this cancer is one option that has been some different groups that has been working in as an option of the treatment of some of cancer cells. And the suggestion is maybe if we could change the metabolism of these dendritic cells during the in vitro production, during the when you are developing these dendritic cells to prepare this vaccine using these antigens from the tumor cells, maybe you can just change the in vitro before to transfer the patients the metabolism of those cells, and to making these dendritic cells more able or more capable to be immunogenic dendritic cells before to transfer to the patients. And the other suggestion is just to inhibit some metabolic or some kinase, for example, AKT, inhibition of PI3 kinase, or some enzymes from glycolysis that can help to maintain this very immunogenic or inflammatory phenotype. And also, so to have this option to change these immune cells in vitro they maybe can be a good option to start to think about metabolic intervention in the response to cancer cells. So this is like how we try to work in our lab in terms of in this cancer scenario. And we started to work with the renal cancer cells because like is this the cancer in Jeff's lab, they are most interesting. But also Karen, my PhD student, she started to work with the glioblastoma. And so the hypothesis of this is our study, it's basically to change the metabolism of these immune cells, as I just showed you guys, as a new approach of cancer treatment. So the hypothesis is the dendritic cells, when they are trying to remove these cancer apoptotic cells, they promote the activation of some metabolic pathway that is not good to keep these cells in a immunogenic phenotype. And so the idea is instead to have a tolerogenic dendritic cells, you can use some pharmacological approaches that is going to change the metabolic pathway that are involved in this to keep these cells tolerogenic, and maybe you can promote the activation of these dendritic cells. So basically, you're going to inhibit the metabolic pathway that we already know are involved to produce these inflammatory mediators, or as well as some molecules that we already know can make the cells quiet, or that are just complicated the situation during the activation of these T cells. So why glioblastoma? This is like one question maybe you guys going to ask. Why you choose like glioblastoma and to start in this? It's not like just glioblastoma. Yeah, you are already working these renal cells. One of the reason is actually we found some really good collaborators, like physicians, that they are basically they are doing this I have 2 hospitals and 3 physicians that they are working in this project, and I can get like better samples compared to renal cancer cells. But the other very important reason is glioblastoma. It's when someone is receiving this diagnosis, they have no more than 1 year and a half month. That so is going to in the median of the survival of these patients, and the treatment's really poor. The treatment nowadays is just this temozolomide, this TMZ, and also the radiotherapy and the surgery. And most of these treatments does not work well. And so this is the reason is to have like we need to have a new approach to help these patients to, have this hope to survive no more than 1 year, or just to have like a quality of life. And so, and this is like the scenario that in the glioblastoma cancer, so you have all these Treg cells in the tumor environment as well. You have these exhausting CTLs that express all this high amount of PD1 that we already know. It's a very immunosuppressed molecules expressed in those cells. You have the dendritic cells. They play a really bad job in this tumor environment. They express this GPDL1 that make these CTL cells in this exhausting scenario. So the idea over here is try to make these dendritic cells that for this accumulation of the apoptotic cells, make these cells in a really bad scenario. So the hypothesis is the glycolysis probably could play another metabolic pathway, could play an important role during the dendritic cells during the efferocytosis of these glioblastoma apoptotic cells. And we want to understand what's going on and how we could inhibit some of this metabolic pathway to make these dendritic cells more immunogenic. So we induce these, use like a cell line to promote like the apoptosis of those cells. And we co-culture these GBM, like, or these apoptotic cells, these glioblastoma apoptotic cells with dendritic cells. And what we observed is the dendritic cells are able to efferocyte these GBM apoptotic cells. As you can see over here, most of them, they have apoptotic cells inside. But what's going to happen when these dendritic cells phagocyte these GBM or these glioblastoma that reinduce the apoptosis? As I told you guys, dendritic cells expressing, just to make like more clear, they express these molecules that we already know are very important to present the antigen and also to promote the cells more capable to activate T cells. But also they express these molecules that can make everything worsen or just to impair the activation. Probably you guys already know, have some immunotherapy that basically block this interaction with the PD-L1 and PD-1 and T cells and dendritic cells, just to impair the exhaustion of these T cells. Moreover, these dendritic cells can also produce these inflammatory mediators, but also can produce these anti-inflammatory mediators during the activation of these dendritic cells. And we are trying to understand what happened when dendritic cells phagocyte these apoptotic cells. And as you can see over here, we don't have a huge difference in terms of expression of MHC class 2, but you do have an increase of CD86. That's good, yeah. So this is, as you know, as I just told you guys, is important to promote the activation of T cells. However, in addition of these CD11.6, these dendritic cells also express a huge amount of this PD-L1 that we already know how bad it can be during the T cells activation. And even worse, the efferocytosis of these GBM apoptotic cells decreasing one of the receptors. We already know it's very important to recruit these cells to the lymph node. So basically, to summarize our findings is when dendritic cells efferocyte these GBM apoptotic cells, they not just impair the dendritic cells to migrate to the lymph nodes, as you can see over here, by decreasing this CCR7, but also they increase the expression of this PD-1/2, that even though some dendritic cells can be recruiting, they are not be capable to activating these T cells. So in addition to that, during the efferocytosis of these GBM apoptotic cells, you have an increase of IL-6, IL-10, and just a slight increase of TGF-beta. So as probably you guys know, this IL-10 and TGF-beta is anti-inflammatory mediators that is very it has been usually are producing in a huge amount in the tumor environment that make everything complicated in terms of activation of macrophages and also Treg cells, but also these dendritic cells can produce lactate and just a little amount of nitric oxide. But when we observe this environment of inflammatory, a mix of inflammatory, anti-inflammatory mediators, we kind of like concerned about this huge amount of IL-6, as you can see over here. So we just went deep in to understand how the cytokines can make this tumor environment even worse. And what we found is IL-6 play a really bad job in the tumor environment. And what they observed is the presence of IL-6 in this is released for the glioblastoma cells. They are related to increase of PD-L1 molecules. And in vitro, what they observed is when you increase the concentration of IL-6, you also increase the PD-L1 expression. So even though sometimes we think IL-6 is a very good inflammatory mediators, we already know that's not true. But in this tumor environment, they make even everything worse, as you can see over here, how IL-6 play a really bad job in this tumor environment. You can see when the animal are knockout for IL-6, you clearly see a decrease of the tumor size and the tumor volume. And when the animals, the IL-6 knockout mice, and we inject, you can see the survival of these animals is much better compared to the animals had still have IL-6. And what we observed is probably this IL-6 could be responsible to inducing this PD-L1 in our dendritic cells and make it and probably is responsible to make these dendritic cells as a tolerogenic phenotype. And the next question that is the hypothesis of this project, and we try to understand which metabolic pathway could be responsible for this phenotype. And using I just brought the results that has been working in terms of metabolic pathway. We have been inhibited to see the metabolism of these dendritic cells. And what we observed when these dendritic cells were treated with 2DG that inhibit basically the glycolysis, and we observed a decrease of CD11.6 that was actually being increasing during the efferocytosis, but also the inhibition of glycolysis impair the expression of PD-L1. That's good, that this helps to make these dendritic cells more immunogenic in immunogenic phenotype, and also are able to increase the CCR7. So basically, summarizing, just inhibit this metabolic pathway, we can promote or can reprogram these dendritic cells to be more immunogenic. And so I'm in the last two slides, just to finish my presentation. We also are interested to understand how this metabolic pathway could play some important role to produce these cytokines. And we observed that when we inhibit glycolysis, it does not change the IL-6 production, but it's it do some effect in IL-10, some effect in TGF-beta, and also lactate production and nitric oxide. And so in addition to glycolysis, we know the fat acid synthase can also play an important role, because this metabolic pathway, it's very important to basically produce the lipid of your membranes, and also to produce the molecules are expressing in the surface of your cells, and the production of these soluble mediators such as cytokines and chemokines. And we use this pharmacological approach and use this synthetic inhibitor of fatty acid synthase. And what we observed, this metabolic pathway seems to be a little important and during the expression of PD-L1, but what we really observed that seems to be very impressive is These pathways are important are involved in the production of IL-6 as well as IL-10. So basically, in summarizing like these results, what we observed is the efferocytosis of these GBM apoptotic cells promoting the expression of PD-L1, that's really bad when you talk about cancer, and decreasing some MHC class 2, and also increases the production of some anti-inflammatory mediators, and that even worsen inhibited the expression of CCR7, that is very important to promote the migration. And the expression and production of these mediators is related to glycolysis and also fat acid synthesis pathway, metabolic pathway, that seems to be important to promote the activation of these tolerogenic dendritic cells. And so trying to inhibit this metabolic pathway maybe can just reprogram these dendritic cells and make these dendritic cells now able to activate T cells. And so just to let you guys know, and you could ask me, so Alexandra, how this can be true, how you can just reprogram the cells, change the metabolism. So this is like one approach that has been used to drive these treatments of some specific cells. You can use some nanoparticles, for example, binding with some antibodies that can drive these nanoparticles straight to some specific cells, for example, dendritic cells. So if you're thinking in the tumor environment, if you want to make sure this metabolic pathway is going to be in some drugs is going to be delivered specific for some specific cells, you can use this approach, for example, in a treatment for cancer that can allow just the dendritic cells being reprogramming in this metabolic pathway. So this is one idea. So this has been using as a good approach to driving a specific treatment for a specific immune cells, for example. So yeah, I think I'm gonna stop my talking now, and I just this is my group nowadays, and I didn't bring like the Leticia's results is about like metabolism and infection cells and infection, especially in Staphylococcus infection, and these results from Karen. Karen is my PhD student, and she's working this glioblastoma model. And also Pedro, it's my dear collaborator, as well as Jeffy, and in this cancer cells, cancer model, and Henrique in the Staphylococcus model. And thank you for, of course, in FAPESP, CAPES, and CNPq. And thank you, Ka, and all, and Gustavo, to invite me one more time to be a part of this course. Okay? So
Great, sure.
I'm glad to take any question. I saw there
Please stop sharing your
presentation.
presentation. So thank you, great lecture. So much important information, and you are very didactic. So students, you are really lucky to have Sean today. And many questions in the chat, but students can start raising hands and asking their questions. Sure, stop sharing the...
Okay, I found now.
yeah,
I was looking for it.
yes.
Yeah, I stopped to sharing. Okay, let's just read this. I saw there was like some question. Okay, those...
But sure. But the
Oh,
students will...
oh, even better. Okay,
Yes, much better.
even better. Who is the first?
Rafa, please start.
First of all, thank you for your presentation. Your slides are so informative and I love it.
Thank you.
I know the focus of our presentation is not exactly about the methodological approach of these techniques, but when you talk about the phagocytosis of apoptotic cells, of dendritic cells, and how these modulate um, the T cell differentiation. I thinking about how this work in the lab. There are three co-culture cells, or you primarily stimulate the cells to induce apoptosis, and after uh,
Good question.
coloca as células
Uh-huh.
dendríticas.
Good
How this
question.
work in the lab?
Yeah, so that's a very interesting question, Rafael. So first of all, like when I start a new project in efferocytosis, like the nightmare, I would say, Rafael, the nightmare is to induce the good quality of these apoptotic cells. And I'm not kidding. Sometimes just take like almost 1 year. Because like the reason, so like just think about what I told you about the non-immunogenic cells and immunogenic cells. If in my apoptotic cell source, if I have some contamination with necrotic cells, is going to be a trouble, because these so I can see like some, like let's say a good effect in terms of activation of dendritic cells or macrophages, but it's not because like it's some my protocol is doing something. It's because I have the necrotic cells as a contamination. So just to try to answer your question, Rafael, the first step of my project is to make sure I have a good quality of apoptotic cells. This means that I don't have any kind of, I have most of my cells, they are apoptotic cells. They don't have any, they are not PI positive, or so they don't have any permeabilization of the membrane. And so this is like, as long as I make sure that I have a good quality of apoptotic cells, then I start, and this can happen in different ways. I can use like drugs to induce like the apoptosis, for example, caspase activators, or in the main protocol in my lab is UVC, that is like more clear. I don't have all these controls. If you use the drugs, you need to make sure the drug is not doing something in your dendritic cells or macrophage. So the second step, Rafael, is to make the co-culture. Is exactly what you said. So I basically culture my dendritic cells, yeah, differentiate with the GM-CSF as the basic protocol everyone have used it, and I do a co-culture. In the proportion that we have done in our lab is always 3-1, but you can find like 5-1, 4-1, but no more than that, because after like more than that can be like not like very useful, because it's just a huge amount, and you can just make your dendritic cells die because you have too much like dying cells. Yeah, and then the third step, Rafael, is just I can some depend of the protocols. I can use like a short time, 6 hours, especially in this metabolism field, and late time in 24 hours, 18 hours. And then I just collect these dendritic cells, and I do all this measurement, like production of cytokines and phenotype of the cells, some functional assays, and yeah. I don't know if I answered your question, Rafael.
Yes, but do you not put the dendritic cells together, the T cells, just the
Oh, yeah.
cytokines?
This is the last step, Rafael. When we decided to work, at least in my field, with dendritic cells, you need to prove, just to show that your dendritic cells produce inflammatory mediators, they express a lot of MHC class 2 or CD is not enough. So you need to make sure they are still like these dendritic cells is really working to do the job you expect dendritic cells do, like to activate T cells. So have different approaches, Rafael, that you can use. 10 years ago, no, I'd say 5 years ago, I could just take the supernatant from this co-culture, and then just do the differentiation of CD4 T cells that I'm interested in my lab using this conditional medium, you know, like it basically is just using this supernatant have been produced during the co-culture. And then I do like a CD4 T cells differentiation using this conditional medium, CD3 and CD28 antibodies. But now this is not enough. Now I need to do like some co-culture with the dendritic cells. But like, give me idea, Rafael, just help me to, you know, like it's super rare to make sure like some compounds of the cells that are the apoptotic cells are degrading are going to be able to activate one specific CD4 T cells. Are you agree?
Yes.
It's
Yes.
super hard, yeah. And so how you have done that before because the reviews asked us like a long time ago. But now what we are doing, Rafael, is what we do is you these apoptotic cells, you just like try to co-culture with the ovalbumin, and then and then I use an OT2 mice, T cells from OT2 mice, and then I co-culture with these dendritic cells. And these allow me to, you know, these dendritic cells going to degrade these apoptotic cells that like they have ovalbumin like inside of these cells, and they are going to present. And then I have the differentiation or not, yeah, depend of the system I'm talking about. So now is the way we are doing in our lab. Actually, what we are trying to do instead just to do this, we say like osmotic shocking, you know, like we basically incorporate ovalbumin in these apoptotic cells. But the other way that you can do is transfect ova in these cancer cells. And this is what we have been working in our lab right now, you know, because it's very important to prove this T cell differentiation. Really good question, Rafael.
Thank you so much.
Perfect. Igor?
Thank you for the lecture.
Thank
That
you.
was very didactic. And my question is, those signals to find me was completely depleted in cancer cells, or they just change to others? Thinking in scenario without treatment.
Okay, good question. I don't know if if I have, I don't know, to be honest. Like I think the finding signals, they are still there. And so the problem is when they phagocyte these apoptotic cells, they are going to make this really trouble, you know, because these cells are going to macrophages and as well dendritic cells. They are going to be in this very anti-inflammatory or tolerogenic scenario. So the finding signals for sure are there because the cells are dying. But the problem is, in this case, the efferocytosis just are making everything even worse. You know what I mean? So I don't know if I understood in this way your question. Who's asking? Sorry.
Me,
Igor.
Igor.
Igor. Did you
Yeah,
understand?
but
Igor,
I'm
yes.
thinking in the pathway of the immune system cells identify and kill cancer cells, sorry.
Mm-hmm. Yeah, in this case, Igor, it's exactly what is not happening, you know. So all these steps that I told you in the beginning of my presentation probably are happening like in a good way, but like the problem is the efferocytosis in this scenario is just like making a very mistake and just impaired what. So the tumor environment already like make all these T cells like exhaust because the cancer cells already can produce some compounds that does not allow dendritic cells, macrophages, and T cells being activated in the right way. And yeah, this efferocytosis just make everything even worse. Yeah, this is like what we know so far. But the efferocytosis happen like in a very well way. I don't know if I answer your question, Igor.
Yeah, for sure. Thank you.
Thank you.
Guilherme?
So first of all, thanks for the presentation. It was amazing. And considering the efferocytosis of infected cells, I'd like to know if is there any case that the pathogen, an intracellular pathogen, can avoid the effects of the efferocytosis and use the efferocytosis as a way for increasing the dissemination, for example?
Dissemination of cells, you're saying?
Yeah, dissemination of the pathogen itself. Yeah.
You know, it's a good question, Guilherme. So what I can tell you is what we have done in terms of metabolism. What we observed is, because always when you work with like infected apoptotic cells, the control that's very important. You have two controls that you need to make sure we're going to run in the same experiments. One of them is just apoptotic cells by themselves without any infection. And the other one is the pathogen you are working, you know. And so what you have seen is when the pathogen it's inside of the cells, in some situation, for example, in MRSA, that's a Staphylococcus infection, when the MRSA are inside of the cells, the activation of the dendritic cells is even better. You know, Guilherme, it's kind of like a Trojan, a cavalo de Troia. So, yeah, so this is like in different ways. Yeah, so when the MRSA are infected, because MRSA, you know, MRSA, Staphylococcus is a very smart bacteria and they have different like escape mechanism. And what we have learned is the efferocytosis in this case, it's very helpful to clear, like during the clearance of this pathogen. So in terms of metabolism, what we saw, we have like less ROS, and so you have less nitric oxide production when you have like when the MRSA are infected directly the dendritic cells. So we have less expression of some MHC class 2 as well, CD11.6. And on the other hand, when the MRSA are inside of the apoptotic cells, everything is much better in terms of dendritic cells activation. So this is like one good, like very well example, like a very good example to show, because sometimes they ask like Alexandra, and I totally agree, you know, guys, like it's important to understand that. So Alexandra, this scenario, and that you have, so just tell me one scenario that you have just apoptotic cells. Does not exist, yeah? This is not true. In the real life, you're always gonna have a lot of apoptotic cells or infected apoptotic cells plus the pathogen. But I think this is the idea, you know, like to make sure these infected apoptotic cells, in case the pathogen can escape for some like mechanism, the infected apoptotic cells just preventing this escape of immune cells activation, you know, that the pathogens can play a very important role. Okay, Guilherme, I don't know if you have a question. Oh,
Yeah,
in this scenario, I don't remember, but I think we've already discussed this topic. But did you compare the dendritic cells that receive apoptotic cells plus the pathogen with dendritic cells that receive the infected apoptotic cells?
Yeah, if I compare, so I need you to run all in the same time, Ka.
All in the same time.
All in the same time. Yeah, so just to make sure, like no, it's not just to make sure, just to to see the difference, like when the pathogens are outside of the apoptotic cells, you know what I mean? And inside
yeah,
of the apoptotic cells. And so,
yeah.
and for sure, but this is not true for all pathogens. For example, for Zika virus, let's say this example, Zika
Mhm.
virus cannot infect the dendritic cells, at least in mice, you know. But when the Zika virus are inside of apoptotic cells, they can make a huge, you know, like trouble in terms of
Mhm.
activation of,
Mhm.
because they, first of all, they can infect, because in
Mhm.
some way they can start to replicate inside of these dendritic cells. Once, like now, they can get inside of these dendritic cells that in the beginning they are not able to. And so in some infections, apoptotic cells can make can you know, like just make the resolution or to establish the infection can be even worse. Yeah, so this is the reason I think in always we need to run in the same experiments, like all conditions, to make sure in the infection directly in the immune cells can be or not different when the phagocyte disinfect the apoptotic cells.
Yes, but I mean, because I think, of course, there is a very complex scenario because we have many stuffs happening together. But when you have, for example, toll-like receptors activation or inflammasome activation or so on, other pattern recognition receptors activation by the pathogen itself, and these cells also receive a different signal for efferocytosis.
You are saying, oh, sorry, you are saying if I do infection
That's
and apoptotic cells at the same time.
yes.
I have never done this before. Always
Because
when I run the experiments, I think it's a very fair comment, and yeah, because in the real life, this is what happened, yeah. So yeah,
yes,
I have never done, and one of the reason is when you have like apoptotic cells and the pathogen, first of
that's
all,
it.
yeah, so I need to try that, and I think the last time I told exactly the same to you. And so
Yes, I remember. I
yeah, I think,
remember that.
and
Yeah.
I haven't done even now, but I think it's really good. I don't know if this dendritic cell is going to survive, but so even though, yeah, so because I needed to make sure like the amount of the number, the ratio that I need to use for apoptotic cells and dendritic cells and the pathogen dendritic cells. I'm kind of afraid that the cells can die, but so anyway, I
I
need
don't
to
know.
try.
Because my
Yeah,
hypothesis is that the efferocytosis could reprogramming these cells not only
yeah,
metabolically in terms of metabolism, but in terms of inflammatory or anti-inflammatory way.
inflammatory. Yeah.
Yes, and this
Yeah.
could be a very interesting to
Yeah, and just think about what you just told me, and if you have the infection at the same time, maybe this can even improve the efferocytosis,
that's
because
it.
the cells can increase the expression of these efferocytic receptors and can make even better. And let's just be honest, yeah? So just think about the steps. So first of all, you have the infection. And then you have all these infected apoptotic cells. And so this is the reason maybe I can put like, I can add like the pathogen a little bit before, and then the infected apoptotic cells that can be even more closer to the real scenario. Yeah, I'm gonna just take a note, because probably I said the same, and I think this is a very important. So for some reason, the reviews never ask for, but so thank God.
I completely understand.
Thank
Yes.
God. But so yeah, I think it's very important to do that. And you know, like just play around and do like put together microorganisms first and then apoptotic cells. I think it's a good idea.
That's it, yes.
Yeah.
So let's move on. Leticia, we have some questions. Still have some
Hi,
questions. Leticia
Leticia.
Saverio.
Yes, hello. Thank you for your class. It's very, very interesting. I like too much. And I have two questions. The first one is about the beginning of your presentation, that it's about the CD47. So I never heard before about CD47, so it's new for me. And I researched about the topic, and I saw that many tumors overexpress the CD47. And I saw some clinical trials that promote the blockade of CD47.
Good idea.
But I have a question that why we don't, why we heard less about the CD47 blockade in compared to another blockade points? I don't know if they are already approved for clinical use.
So the antibody against CD47, are you asking me? Yeah, I think
Yes.
they have some in clinical trial, Leticia. And so this is like, you know, so the CD47, as I told you, the same, remember the same I showed for arteriosclerosis diseases in the beginning of my presentation?
I think, yeah, I
Yes,
think so.
in the arteriosclerosis, like the cells overexpress the CD47, and this allows you, so the probability of these patients to have like a heart attack or stroke, it's really high. And on top of that, they have some deficiency of the efferocytic receptors. And for cancer, I just like when I was preparing this presentation, I thought like I brought these results, but for some reason I decided to remove it. There is this clinical trial using this antibody. And just to think about that, yeah, just if you block these don't eat me signals, you are able to phagocyte. And just like I'm showing you guys the bad side of efferocytosis in cancer, but this needs to happen anyway. Because how your dendritic cells, you know, like the vaccine for cancer is basically this one, you know, you just try to grab all these tumor antigen and give to your dendritic cells, to phagocytes, to, you know, capture all these antigens. And efferocytosis is the best way to do that. But the problem is, you know, first of all, like I think I don't remember it, I think it's a breast cancer, and I think I saw something in prostate. Have you seen
Yes,
yeah.
I see the prostate cancer.
Prostate is very common, like to overexpress this CD47, you know, and in some way, it's a way the tumor can just impair, you know, to be removed and just keep, like it saved some time, and then they can proliferate more and more and just avoid some, the presentation of their own antigens, yeah? So this is what cancer does not want at all, you know, to have some samples of itself to present T cells. Yeah, so it's a good way for sure, and it's I don't know if like it's very successful this treatment, but so as long as I know, it's one approach that they have been working in terms of to promote the ferrocytosis, yeah, blocking these don't eat me signals. Good, like
Pretty cool, thank you.
Yeah.
And I have the second question that it's about dendritic cell in glioblastoma model. I don't know if I lost the parts of your presentation that you talked about, but my question is, this dendritic cell is from the patients, so it's autologous dendritic cell, or it's healthy donors?
Oh, thank you for this question. So far, Leticia, is just like I'm using cell line, glioblastoma cell line. And so this is like as a source of this glioblastoma cell model. And so the dendritic cells is from bone marrow. It's just like in the beginning of this study. But so just to answer your question, The second part of our study is just to understand how dendritic cells, the behavior of these dendritic cells in the tumor from the patients. So in the total, have 90 patients so far, and we are still learning to work with this cancer tissue actually. But now what I can tell you that it's already described in the literature, and what they observed is actually, and we observed the same in our lab. The monocytes from these patients, they have like a much more PD-L1 expressed in the cell. So it's basically, they already have some immunosuppression in the monocytes are in the blood, yeah, first of all. And what we observe in our dendritic cells that we isolate from the tumor, and these dendritic cells, it's in a really bad shape in terms of activation. You know what I mean? So in our dream that we are learning is, I don't know if this dream is going to be true. So the idea is when we got, so first of all, yeah, just, I don't know if you guys are familiar to work with patients, sample from patients. This is like really new in my lab and new in so many ways. Kai, we have time, yeah, just to share this experience that I think we don't have a lot of
We are on
time,
but go ahead.
Yeah, it's just like to make a long story short, Leticia. When you start a project, you think like, oh, this can be true. But when we start to work in these samples from patients, we are able to separate, like to isolate dendritic cells, and we can see like these dendritic cells, they already have all these no pleasure molecules, for example, high expression of PD-L1. But the comparison is kind of like crazy because you don't have samples to compare from healthy, you know, so what we do when you have just one sample, you have like some patients that they have some, it's not a cancer, you know, like it's kind of like it's not GBM at least. And so we are trying now to compare it, like compare these dendritic cells from these two samples from different patients. Yeah, so this is like what we are working, Leticia. But the problem is these patients does not receive any radiation or the treatment. So the cells are not dying by apoptosis in that stage. But at least we are trying to understand how these dendritic cells are working in this tumor environment. But so just to answer your question, it's all cell lines so far.
Yes, I'm asking this because I work with dendritic cell and glioblastoma, and when I use the dendritic cell, immature dendritic cell, I see the same phenotype that you show it. But when I activate the dendritic cell with TNF alpha, this phenotype changed interestingly, thinking about the anti-tumor therapy. And I use the dendritic cell from healthy donors.
Oh, have to do. Yeah, this is my dream. So I'm trying to collect now these patients and these healthy people, and I do this comparison with healthy people and bone marrow monocytes from patients. Yeah, but so
Great, awesome. Thank you.
Thank you, Leticia.
Thanks, Leticia.
One more Leticia.
Leticia Serafim.
Oi, Leticia.
Hi.
Hi,
There's
how are you?
too many Leticias here. So
many Leticias, yeah.
Thanks for the class, Professor. It was amazing. Actually, I don't work exactly with immunology. I'm from the microbiology department, but so I don't know. I don't understand a lot about these things that are more specific. But I was wondering if it could be possible to induce cells to perform efferocytosis, to use them to treat disease in a way similar to the CAR T cells. I don't know if it's a correct way of thinking, but I was just wondering.
I think during the infection, Leticia, maybe you can improve maybe the efferocytosis and to help, like just improve, for example, the number of the receptors involved in the efferocytosis. In cancer, actually, it should be the opposite. Yeah, so to remove these apoptotic cells, it's in some way just make everything worsen. And the idea, I think it's better to the idea of our approach, just to change the metabolism could be the best way that the dendritic cells and macrophages can remove these cancer cells, and at the same time they can still be activated. And so, but I never thought, like, I don't know if I can do some correlation with CAR T cells. Is just like to improve the efferocytosis or change the metabolism of these cells. Leticia, I can see that can be helpful to clearance the infection or to impair the immunosuppression in the tumor environment. Okay, I don't know if I answer your question.
Yeah, sure. Thank you.
Last question, Amanda.
Amanda?
Hi, and first
Hi,
I
Amanda.
want to thanks for the lecture, amazing lecture, and I want to ask, if can the process of the efferocytosis happen with cells undergoing an inflammatory death process, such as pyroptosis and necroptosis, before their actual death, perhaps as a mechanism to control this inflammatory death?
So actually, just to be clear, the expression efferocytosis nowadays, it has been used for different kind of cell death. Okay, Amanda. So in the beginning, we start to use just for phagocytosis of apoptotic cell, but nowadays we use it like this efferocytosis for any kind of clearance of these different like way to when the cells are dying. And so I don't know if I understood well your question. So we are saying like just if in this, for example, I told you doing in this the efferocytosis of apoptotic cell, this kind like specific cell death, just make everything like very immunosuppression and all these anti-inflammatory mediators. You are asking me if in different kind of cell death you can have different kind of environment. This is your question?
Yes, just like in pyroptosis and necroptosis, it had a inflammatory death.
Inflammatory
So
death.
I want to know if the dendritic cells can identify these receptors, the bind me and find me in this cell before the explosion of the cell and contain this cell in inflammatory process.
You are saying like you can anticipate, for example, in before to these cells, but pyroptosis, like pyroptosis is something like they start to release it in a really short time, all these names,
Necroptosis could be, because necroptosis is, I think, is a longer kinetics, but pyroptosis happens so quickly, and I don't think these find me or it me signals
can happen,
happen before. But it is a very, very interesting question. Could be. So could be a manner to dump down the inflammation caused by these inflammatory cell death processes.
It's dependent of the intensity of the signal. If you talk about Nigericin, obviously not, but the signal is more
mild,
slowly.
yes.
Can be
And of
worse.
course, it also depends on the cell type, because we know that some some cells don't die or die later on, and maybe it could be a very interesting.
Yeah, to know. Yeah, Amanda, I don't know if I can answer your question. So I think it depends like the simulation, and so how fast these cells die in a different way than apoptosis. And I think the finding signals, because it's a different way, these finding signals are released by pannexin channels, different from, for example, pyroptosis, they use gasdermin. So it's the size of the molecules that are being released is totally different. And I think I think it depends of, I think the finding signals are going to be producing, and the macrophages can also phagocyte. This is the reason we use the same efferocytosis for all.
Burns,
Yeah. And so, but what I'm concerned is, I don't know if when they phagocyte these other kind of dead cells, for example, necroptic and cells in pyroptosis, I think can be pretty much the same. But in the phagolysosome, I don't know how fast this degradation is going to happen, and you can promote some activation of these dendritic cells, yeah, or macrophages, yeah. So next time I'm gonna bring some studies in other kind of efferocytosis using other, pyroptosis or necroptosis, and then I'll let you know.
So that's it. We are late now. So thank you, Sha. Thanks a million again. So fantastic.
Thank you guys for all.
And hope to see you in the next year again.
Hopefully, I'm glad to and maybe as I told you in person, and then you can just keeping the class easy, and you can interact with each other. But thank guys for I love the questions, and Kat, thank you again for the invitation. What's amazing to be here one more time. Okay.
Okay, see you. And see you guys.
Bye,
for
guys.
my more two lectures at
Today,
afternoon, okay? See you at 2.